This prediction asks you to update something foundational about how you conceptualize medicine. Every pharmaceutical we currently have, every pill, injection, infusion, is a chemical entity, foreign to the body, administered at a point in time, producing its effect through metabolic interaction, and eventually cleared. The entire architecture of drug development, regulatory approval, dosing science, and pharmacovigilance is built around this model. Synthetic biology proposes to replace that model, in some domains, with something qualitatively different: a living, self-regulating, genetically engineered biological system that resides inside the patient and responds to physiological conditions in real time. The existing proof of concept is not hypothetical. Synlogic, a clinical stage company, has engineered strains of E. coli to produce phenylalanine-degrading enzymes within the gut of patients with phenylketonuria, a metabolic disorder in which phenylalanine accumulates to toxic levels. By engineering bacteria that produce the therapeutic enzyme exactly where and when it is needed in the gut, in response to dietary phenylalanine, the approach eliminates the need for dietary restriction and provides more consistent metabolic control than any pharmaceutical alternative. This is living medicine in clinical trials, not theory. The extension of this platform is where the ambition becomes both exciting and sobering. If bacteria can be engineered to degrade phenylalanine, why not engineer them to produce insulin in response to blood glucose levels, a living, self-regulating pancreatic substitute for type 1 diabetics? Why not bacteria engineered to produce serotonin or dopamine precursors in the gut in response to detected neurotransmitter deficiency signals, a living antidepressant? Why not engineered bacteria that detect the presence of a specific pathogen and produce a targeted antimicrobial molecule on contact, a living antibiotic that cannot produce resistance because it evolves with the target? These are technically plausible directions. Technically plausible within a decade, in some cases. The regulatory and ecological frameworks required to govern them, however, are significantly underdeveloped. The warnings here need to be stated directly. Engineered organisms released into the human gut interact with a complex ecosystem of 38 trillion existing organisms in ways that are not fully predictable. Horizontal gene transfer, the process by which bacteria share genetic material with one another, means that an engineered gene construct introduced in one strain could, in principle, transfer to other gut bacteria with unpredictable consequences. Containment strategies, engineering kill switches into the bacteria, designing auxotrophs that cannot survive outside specific gut conditions, are being developed, but the complexity of gut ecology makes absolute assurance impossible. Ecological safety extends beyond the individual patient. What happens when a patient with engineered gut bacteria encounters antibiotic treatment? What happens with fecal excretion of engineered organisms? These questions need regulatory frameworks before the technology reaches widespread deployment. The science is extraordinary. The governance infrastructure must match its ambition.